And these are obviously a very functionally important part of the fungus, involved in the sort of reproductory process of the fungus. But actually, the main component of the fungus, the most functionally important part of the fungus, is occurring below the surface. This huge mycelial network of mycelial cords that interconnect forest trees, and they go throughout grasslands and natural ecosystems. And these fungi, these hyphae, are responsible for the distribution of nutrients throughout their ecosystems, and they're ultimately, most importantly, responsible for the degradation of organic material. They convert dead organic material into nutrients that can be taken up by other organisms, but through this process, they determine how much of the carbon is released into the atmosphere and how much is ultimately stored in the ecosystem. These fungi physically give soil its structure. They bind aggregates of soil together, and they form microhabitats that other organisms can live in. They keep the soil a living thing, ultimately. And even the decomposition of that fungal necromass, when the fungi die, the decomposition of that necromass is the process through which soil organic matter is actually formed. So these fungi really determine the balance of carbon between the soil and the atmosphere. And even though we know different fungal groups will do these things at completely different rates and in completely different ways. One fungus will decompose wood 100 times faster than another fungus, and yet we know nothing about which individuals live where. And that's primarily, it's probably driven by the hyper-complex nature of fungal communities, which has sort of forced us down the line of having to use genetic sequencing approaches to characterize their global distributions. So the majority of global-scale fungal ecology studies have used genetic sequencing approaches to try and characterize all the thousands of species that live in each patch around the world. And what they consistently find is that the further away you get, the more dissimilar the fungal communities get. So the further away you are, the more different combinations of species you get. And this has led some people to assume that fungi are somehow fundamentally unique. They're different from all the other organisms in their biogeography because there are no meaningful patterns in their biogeography. You don't see clustering of some species in some regions and clustering of other species in other regions. Now, like fine-scale microbiologists that are focused on individual regions will say that there's plenty of evidence for that, but the global patterns never seem to provide any evidence for clustering of fungal groups. But I actually think if we applied the approaches that we use to study fungi to plants, we would see a very similar thing. I've got here just a basic image of the general biomes that we see for plants, that is, they're indicating different plant types. And we can all of us know the difference between a desert plant and a forest plant, per se. But if we looked at these plants the same way we study fungi, if we just generically sequenced them and identified their species composition, the species that are existing in the desert in Australia are taxonomically very distinct from the species that are existing in the desert in North America. It's only when we look at their functional attributes, their traits, that we really see the patterns. If we simply looked at the species composition, it would be much more difficult to see the meaningful patterns. But when we look at the characteristics of the organisms that allow them to live in those ecosystems, we can start to see broad biogeographic patterns. Again, all of us can tell what a grassland plant looks like next to a forest plant. We all know what a tree is compared to a cactus. And these are the kind of basic bits of information we want to try and generate by using a trait-based approach to understand fungi. And so what we did is we collected fungi we were sent fungi from all over North America. And they were a range of different species from a range of different phylogenetic groups, but ultimately they were all functionally equivalent wood decomposition fungi. So they govern the rate-limiting steps in the decomposition of wood. And they release carbon from decaying organic material, and they release it into the atmosphere and also sequester some of it in the soil. And so we have the full phylogenetic history of these individuals, but they were a random subsample of fungi from across North America. These are very common, ubiquitous species, and they grow in a lot of different places. And if they were growing in the same place, they would be competing with one another to sort of colonize wood. But ultimately, they were all collected from different places. And what we wanted to do is simply look at their hyphae, look at those fungal networks to see if we can see any characteristics of those hyphae that can help us to discern which ones are from the dry regions and which ones are from the wet regions, which ones are from the Arctic and which ones are from the tropics, because we had a huge climatic variation. So We actually measured a wide range of functional traits on these fungi. We measured their growth rate, their morphology, their chemical composition. We measured their enzyme production and their volatile organic compound production and their cell structures, all sorts of things that might help us to discern where they come from. And the most important, I'm going to focus on some of the most important traits that we measured during this process. And actually, this is one that we thought would be our best predictor of where the fungi come from. So we measured, this is just a physiological growth response curve. So we grow each of our fungi along a range of different temperatures, and then we simply measure how quickly they grow at each of those temperatures. And we thought, okay, this is going to be great. We'll grow them at all the temperatures. The fungi from the tropics will grow optimally under 30 degrees, and the fungi from the Arctic will grow optimally over here, and the point at which they grow best will tell us where they come from. There's a trait that relates to the environment. And so we grew all of our fungi across a range of environments, and we found our initial theory was entirely wrong. Everyone wants to live in the tropics. All the fungi that we had, whether they're from the Arctic or the tropics, they all wanted to live between 25 and 35 degrees. So we thought, okay, either we've failed or they don't use temperature as a selective, maybe it's not a very good selective pressure. Maybe they're living under the soil and they don't really care about temperature. So we thought we'd do it over a gradient of moisture, because that's a much stronger selection pressure, and we found exactly the same thing. So all of our fungi they grew most optimally under moist and warm conditions. Everyone wants to live in the tropics. But as we explored the data set further, we realized the most meaningful axis of variation isn't the highest, the maximum place where they grow, the fastest growth rate. It's actually their niche width. So a lot of the fungi had very narrow niche widths, so that's a very narrow hump, and they can't survive under very dry conditions, whereas others can grow under incredibly dry conditions. And we think this variation maybe is something meaningful. But it makes us think the individuals that are growing here can grow everywhere, whereas these individuals simply couldn't exist here. So for these ones to exist, there must necessarily be some sort of trade-off that allows these ones to be incredibly competitive under the optimal conditions. Otherwise, they just wouldn't exist anywhere. And so this is a concept of a trade-off, a dominance-tolerance trade-off, which is certainly not a new concept in community ecology. It's a very old concept. The idea is simply that your ability to do something or to express a trait necessarily comes at the expense of your ability to do something else. And so we sort of proposed based on this that the individuals that are able to live in the tropics are the ones that are able to fight, while the individuals that are living in the desert are simply able to tolerate stress. And the ability to tolerate stress comes at the expense of your ability to fight. But the only way for us to find this out was to fight all of our fungi against one another. So this was about 1,600 fungal combinations. It took a hell of a lot of time, and a lot of people lined up doing Petri dishes in our lab. But ultimately, it was a fun process because they made loads of pretty colors and did loads of interesting things. And we measured the growth rate towards one another and the growth rate over one another and under one another. And we ultimately used a range of different approaches to characterize which individuals had won. And this allows us to come up with a ranking. And finally, I was delighted to find that one of our theories was correct, that actually we do find evidence for this dominance-tolerance trade-off. So that's simply saying the individuals that are highly competitive are very bad at tolerating climate stress, whereas the individuals that are stress tolerant, they are very non-competitive under optimal conditions. So this certainly isn't the first time anyone's shown a dominance-tolerance trade-off in a group of organisms, but it is one of the first times that it's been shown at such a broad spatial scale, and certainly with a group of fungi. And the reason that this is possibly the most exciting graph that I've generated during my research is because I find it fascinating that we've got a fungus that's growing in a log up in Alaska, and another fungus growing in another log in the tropics, and they're ultimately governed, their distribution, their existence where they are, is governed by this fundamental relationship, this fundamental trade-off that is acting across both of them. They're both either, the one in the Arctic is unable to be competitive because of its ability to tolerate harsh conditions, and the one in the tropics is highly competitive despite its ability to not tolerate harsh conditions. Sorry. And so when we throw all of this information into a principal component analysis, this is a different kind of graph that essentially it allows us to explore the differences between all of our fungi. And if we throw all of our traits in, I realize it's a bit of a complicated graph, but we're just trying to see which sort of traits correlate with the differences in our fungi. When we include all of our 157 measured traits, this dominance-tolerance trade-off comes out as the most important axis of variation. So the individuals on the right are the fast-growing, they grow optimally under optimal conditions, they are highly competitive, whereas the individuals to the left are stress-tolerant. They can survive under cold and dry conditions. And this dominance-tolerance trade-off correlated perfectly with a well-known trade-off in growth ability. So if you imagine as a fungal hyphae, you grow by producing tubes. And so you can either grow your tubes very long and fast into one direction, or you can grow them all on top of one another. And the individuals over here are investing all of their growth on top of one another. So they're growing sort of thick, dense mats, where the individuals over here are growing fine tubes. And this growth difference seems to be underpinning everything that we're seeing. So the fungi to the left is a sort of exemplified by this fungus here. It's very thick and dense, and it's got a buildup of hyphae that's protecting it from the harsh environment. But that by allocating all of its growth on top of itself, it can't extend very rapidly. Whereas the individual on the right is growing incredibly rapidly, and it's growing over its opponent and destroying its stress-tolerant competitor. So these simple traits seem to be explaining some patterns underpinning these organisms. But the relevance of this comes when we link it back to the environments that they were collected from. If you remember, we've got fungi in the Arctic and fungi in the tropics, and we're trying to tell which ones go where. And if we look at any of the individual traits, any of the single traits on their own that we've measured, for example, here, the climate stress tolerance would be the temperature niche width. They don't correlate with any of our environmental characteristics. We know all sorts of things about the environments that they were collected from. We know the temperature, the moisture, the soil characteristics, all of these things. We can't predict anything based on individual traits. But when we convert that trait to a dominance-tolerance trade-off, so simply divide the competitive ability by the climate stress tolerance, finally it sort of stretches out our axes and we start to make meaningful sense. We start to see method in the madness. There's a pattern here. We can see in the warm climates, these are the competitive ones relative to their tolerance ability. And it wasn't just temperature that correlates with this trait, this new trait. We also had the aridity of the soil, the amount of moisture available in the soil, the texture of the soil, the pH of the soil, and the organic matter content all work together to sort of structure this dominance tolerance trait. And because we know all of this information at every single pixel across North America, we can generate a spatially explicit map of this dominance tolerance trait. This physiological trait, we can finally say some characteristics of these organisms that allow them to exist in one place rather than another. And we can finally say the individuals in the red parts are more competitive and the individuals in the white parts are more stress tolerant. So this basic functional biogeographic information is useful. It's very interesting for us to understand where fungi exist, but it's most valuable when we link this trade-off, this trait, to the actual functional attributes, the attributes that tell us how they are affecting the carbon cycle. So we've got a postdoc in our lab, Nikki Lustenhauer, at the moment, who's currently exploring that, looking at how this dominance tolerance trait relates to the wood decomposition by each of those fungi. So we've grown all of our fungi on little blocks of wood, and we simply measure how quickly those bits of wood lose mass. And that's the decomposition rate. And we find very strongly that the individuals with a high dominance tolerance, the competitive, fast-growing ones, the thin tubes, those are the individuals that are decomposing very rapidly. They decompose that wood fast. Whereas the individuals that are growing slowly and stress-tolerant individuals, they don't invest much in decomposition, so they're growing very much more slowly. And so because of this simple linear relationship, our functional biogeography map can turn into a map of intrinsic fungal decomposition rate. We can say the fungi here are fast decomposers and the fungi here are slow decomposers. And even more interestingly to us is that the fungal structure, the hyphal structure, is even different between these groups. The individuals up in harsh, cold, difficult conditions, they invest a lot in these calcium oxalate crystals. So there's these jagged little crystals across all of the hyphae that are they're sort of protecting the hyphae from the outside world, and they're also making it a very harsh, recalcitrant structure. Whereas the individuals from the tropics are all fine, beautiful, thin tubes with no calcium oxalate along them. And they actually have a low carbon to nitrogen ratio, so they're very easily decomposed. So when these fungi die, these ones are incorporated much more efficiently into the soil carbon, whereas these ones are decomposed very inefficiently, and a lot of the carbon is released back into the atmosphere. So the structure of this dominance tolerance trait is starting to tell us not only how these fungi govern carbon loss from the ecosystem, but also the carbon stabilization within the soil. So this is obviously the